Right, chapter 3 in our book, our next chapter that we're going through is called Measuring Color. Start looking at color and how we measure it. Start with basic measurement. If we want to work with colors, we must be able to quantify those colors. So somehow we have to measure them. Okay? We can do it either with our eyes and our brain. That's, believe it or not, a way of measuring it. Or we can do it with instruments. So we somehow we have to measure that color. Color measurement is a special technique of analysis. The key is when we do it, we should do it consistently the same all the time. Whether we do it visually or using instruments, we should be consistent with what we're doing. So like most scientific measurement, accurate color measurement is dependent on obtaining a representative sample. So the sample that you are measuring has to be a good sample. It has to be representative of what you want to quantify or to measure. And there's something called the first law of analytical chemistry. Probably all familiar with this since you all love chemistry so much. That analysis is no better than the sample. So if you have a poor sample, you're not going to make a good evaluation of it. And in color, when we make our samples, we generally do it on an injection molding machine. So if we do a poor job of mixing, or we get the sample dirty, or it's got specks on it, that's not a very good sample. It's hard to get a good reading on it. And the second law of analytical chemistry, the analysis is no better than the standard itself. What we're comparing our sample to needs to be a good representative sample also. Sometimes we see in industry that the standard has some problems too. Maybe it faded, or maybe it's not even of the right color that the customer wants. So we have to be careful with that. Like I mentioned, two main types of measurements. The first one is visual. Remember, that's the way we all normally look at color. So it takes the eye-brain combination along with our optic nerve and all those cells in between, and we say that our eyes are a good null detector. It's good at detecting whether a sample either matches or doesn't match. So when we look at standard in the sample, we can say, yep, that's a good match, or no, that's not quite right. When we were doing those exercises in the book, when we got all the chips in the right place, we could look at the whole thing and say, yeah, those look pretty good. Or if there was one out of place, you can pick it out. The hard part for us to do is we're not as quantitatively as good, which means we can't tell how far off it is as easily or which direction is off, especially, say, the average person. Now, if you're used to working with color all the time, you can do that more accurately because you're used to doing it. It's easy for the average person to say, nah, that's not the right color, but I don't know if it's too red or too green or too light or too dark, or to quantify that difference in terms of those attributes we talked about in that Munsell system, hue, value, and lightness. How is it off and by how much too. And here's just a picture from your book of how we do visual measurement. But actually it has one thing wrong. What's wrong with this as far as looking at samples? What is that person doing wrong when trying to see if that standard and batch is the same? Yeah, exactly. The samples should be touching. Because if there's a gap in between, then it's difficult for us to see if there's really a difference. That's why if you're painting your car, or you get a dent in your door, it's better to paint the whole door than to touch up a section of it, because it's easier to see the touch-up paint if it's right against the base paint than if there's a line around it, like where the door separates from the quarter panel. You can't notice any difference as easily. The other type of measurement is using instruments. So we have some sort of electronic detector computer combination. It does a better job of quantifying how far off we are in which direction. You can get numbers that tell you how far the color is off from your standard as your sample. You can say which direction. It'll tell you it's so much too red, so much too blue, so much darker, and give you numbers that describes that. The problem is it doesn't always agree with visual results. It always gives you numbers, but those numbers may not agree with what you see visually. Most of the time, they're pretty accurate, but in some cases, they're not. And again, we're trying to model our visual system using instruments, and it doesn't always work exactly like our eyes work. You got to be careful that you take the readings from the instrument kind of with a grain of salt and make sure you look at it. You always have to look at the sample. There's two main types of instruments that are used for measurement in industry. One is a tri-stimulus colorimeter, and the other one is a spectrophotometer, or sometimes called a spectrocolorimeter. The colorimeters are much less expensive. The tri-stimulus colorimeter, they'll be used mainly for quality control. The spectrophotometers are what we have in our lab. They're much more expensive, much more sophisticated, and they give you more information about the colors that we're looking at. Colorimeters directly measure CIE colorimetric coordinates. So you get numbers out of it, basically. And that's all you get. And there are only certain numbers you get. And it uses 3 filters along with a single light source. And it looks pretty much like this. See your light on the top there shines on a 45-degree angle through a slit. We have 3 filters that rotate, and then you have a detector, and then it gives you numbers at the end. So it's basically just looking at numbers generated by light going through 3 filters. Fairly basic, fairly simple compared to a spectrophotometer. Spectros analyze light energy reflected or transmitted by the sample, wavelength by wavelength. So it doesn't just look at 3 specific colors, but it looks at all the wavelengths across the spectrum. And actually, it doesn't look at every single one of them. It goes in generally 20 nanometer increments. The real fancy ones will do every wavelength, but usually they're about 20 nanometer increments. So like when we did our calculations, did it on the board here at 400, 500, 550. In a spectrophotometer, they would have a point every 20 nanometers. So at 400, 420, 440. So it'd be more accurate of a curve that it could draw. Because one of the things about spectrophotometers is that it can produce a curve that we can look at and identify the color with. Here's a picture from your book that shows the parts of a spectrophotometer and what you get from it. We have our sample here. Light source shines light, and this happens to be an integrating sphere type spectrophotometer that we'll discuss a little bit later. But the light hits the sphere, hits the sample, and then goes, in this case it shows a prism. It breaks the light into individual wavelengths. This slit moves, and then it records how much light of each wavelength is present. And then it records that, and then it gives a graph of how much light it detected at each wavelength across the spectrum from 380 to 780. There's also diffraction grading as another way of doing this instead of a prism. Prism was done in the very early spectrophotometers. Now they don't use actual prisms, they use other devices. Slide that shows the difference between spectrophotometers and colorimeters. Spectrophotometer on top. You can get a reflectance curve, multiply it by the illuminant and the observer function, and we can convert that to x, y, and z values, and then we can convert it to all the numbers we'll talk about later in the course. I'm sure you're all as anxious as can be to get to this mathematical part of it. The colorimeter has the light source, the filters, and it goes directly from the tri-stimulus values to all these numbers. There are no curves. It doesn't give you the spectral curve, doesn't give you an object curve, so it doesn't help you identify what the pigments are. Mainly used for quality control to say if a color is right or not. So a colorimeter only has a 2-degree standard observer. Remember last time we talked about possibility of having a 2-degree observer or a 10-degree observer. Those are the 2 types of observers. 10-degree observer is a bigger area. Only can use illuminant C, used for quality inspection, and gives colorimetric data only. So it only gives numbers it doesn't give curves. So it's much less flexible than the spectrophotometer, which gives you a choice of observer. You can have any observer you can think of in there. Also, any illuminant. You can have all real luminance or even just calculated luminance. You can use it for quality inspection, but also for R&D, and for our purposes we call R&D color matching, to develop formulas, and gives both spectral and colorimetric data. Spectral is the curves, colorimetric is the numbers. When we work with instrumental color measurement, we're taking the subjective eye-brain combination and replacing it by a sophisticated instrument and a high-speed computer. And we're trying to model the real systems with what make up and try to take some of the subjectivity out of it. Now, the instruments are much better now than they were 10 or 20 years ago, that they are very repeatable, reproducible, and accurate. That's a good thing. The question is, are visual color measurements still important? Have instrument color measurements advanced to a state that visual measurements are no longer needed? Are visual measurements subordinate to instrumental ones? And are the uncertainty of visual measurements so large that for practical color technology, only instrument measurements should be used? You go to a plant, you have several people that are making color decisions. You know, there might be a different quality person on every shift. There's some difference between the way they look at them. You know, they're always looking at it the same. Or, you know, you're just talking about all the stuff you're doing and you had 1 hour sleep last night or in the last 24 hours or whatever it is. You know, if somebody's working midnight shift and they've been up for 2 days straight and now they have to make color measurement decisions or color decisions, how well are they looking at that sample? You know, are they tired? Are their eyes working as well as they can? Is somebody in a bad mood? All those things can affect it. Remember, our night instructor, Bob Charvat, had a guy in one of his classes. Early in the term, they did the Munsell 100 hue test, and the guy got a perfect score. He got 0 wrong. And then at the end, on the finals day, Bob had them all do it again, and this guy came in an hour late. He been at work and a lot of problems at work, so he's all flustered, all stressed out coming in. Came in, did the test. He got like a 50 on it, much higher score. And it's the same person, just his state of mind was different. There's a lot of variations when people do anything, really. Are we better off just using instruments that are repeatable and always do the same thing? So that's some of the questions we'll try to answer as we do this entertaining presentation. Let's start with the basics of measuring color. First question is, why are we making the measurement? What are we after? Are we there or doing it to define its appearance, to determine if it matches another color? So are we doing a color match? Or to determine its composition? Do we want to reproduce it by mixing up colorants so that we get the same color match? So if we're just measuring appearance, we just have our sample and we just look at it under whatever light source that we want to look at it. Showed this before. Do 2 samples match? You have to have your 2 samples. And the best way to determine a color's composition is to use a spectrophotometer and look at the curve. The curve can be very useful to us for deciding or determining which colorants are used to make up that sample. So in the lab, we have several books of our individual colorants that have the curves associated with it, so we can look at the curve of an individual colorant, look at a mixture, and decide what particular colorants are in there. And actually later in the term, we will do that as an exercise. I'll give you several individual curves for individual colorants, and then you'll look at a mixture and be able to tell which colorants made up that mixture. So when measuring color, we are really performing 2 basic functions. We have to examine the color, and then we need to make some kind of assessment to it. So first thing is to really just look at it and look at it properly so we can make the correct assessment of is it good or is it not good. So our components of examination, we have to have our light source, sample and standard, and our detector observer. Hopefully by now you have this burned into your mind that we said it enough times. To see color, you got to have a light source, a sample, or an object, and then some kind of detector. First detector we'll investigate is our eyes. Then we'll look at instruments. First component of assessing whether or what we're looking at is there a difference? You look at it, remember I said our eyes are good null detectors. So the first question is there a difference between the standard and the sample? Then we need to describe that difference somehow. And we could describe it by words, or we can describe it by numbers, whatever we want to do. Sometimes words are good enough. Sometimes they're not precise enough, so we need the numbers. And then finally, the ultimate question is, if you're in an industry setting, is the sample acceptable or rejected? We could have a sample and a standard that we can say, yes, there's a difference, but the difference is small enough where it's still an acceptable match. So if we're making a match for a cheap toy, we don't need to be quite as close as if we're making a match for automobile application. That should be a much closer match. We were talking earlier about the door and the other panel not matching, door and the dashboard not matching. Ideally, that's supposed to match. One of the problems is the door panel is probably made out of a different material than the dashboard is. Is that in that case the same? Different machines, though. Certainly different molds. And the question is it the same material that goes into both? There's a lot of variables that go into it. Even it could be the same lot of material and everything's the same, but maybe the conditions are different. That can change it. The grain, like you say, can change it. We'll see an example of that a little bit later. So there's a lot of things involved on what causes the difference. The question is that difference acceptable or not? That's a subjective decision sometimes. Sometimes we'll see later on when we talk about instruments, there's numbers involved. We can have specifications for how close the numbers have to be, but there's problems with using numbers. There's problems with everything, I guess. But, there's some problems with visual and there's problems with numbers also. The question is, which one do we use? So first thing, we'll look at the example.